So welcome to this presentation where I will present the basics about the REC data. And this is the data that you need for your research. So I'm going to talk about the RECBASE. And my objective here is going to be to talk about the basics. I will give you a short overview. I will talk about some details about the data, the structure, the time frame of the data sets, the spatial coverage, etc., so that you have an idea of what we have in our database. Well, first of all, I want to tell you that we don't actually talk about the REC database. We talk about something much shorter and nicer, which is the RECBASE, which is an acronym that we made so that it's sort of nicer to talk about the database. The collection. As you know, REC itself doesn't collect any data. We get data from data providers. So how many and what are those data providers? Well, we have 4 data providers. We have OpenAcademy, we have Syke, we have Luke, and we have Logos. And most of the data sets come from these 3 data providers, Syke, Luke, and Logos. From OpenAcademy, we already have 1 data set, and it's very local. I will tell you later. So when was this data collected? This data collection was made in 2 years, mainly in 2 years, from 2018 to 2020. I came here at the beginning of 2020, so most of the work of the data providers giving or providing the data was done in these 2 years. I was not here when all the work was done, or all the huge work was done. All the work was done by Bess Hardwick, and then also Tanya Lindholm, because Bess Hardwick was on maternity leave around here, so Tanya was replacing her. It's in the P drive, and actually it's nice that you know that it's in our internal P drive, but if you need any data set, if you need any extract from the data, you just tell me. That's what I'm here for. The structure. What is the structure of this database? How many fields? What do they mean? Well, the database consists of 22 fields. I'm going to tell you which ones. This is the list of all the fields, and these fields they have some basic information. We have the coordinates, we have the time, very important, when it started, when it ended. We have also some information about the sample, about the species, and about abundance, effort type, etc. Another way of seeing this is if I make them in several boxes, if I classify the fields in several categories. So we have the basic info, we have the geographic information, we have the time when it started, when each observation started, when each observation ended, some information about the sample, the data type, event, species. Species is actually a concatenation of this and this, and then information about abundance, effort, and effort type. And how many data sets do we have? Let's count it. Let's go. How many? 1, 2, 3. Ooh, it's going faster. 15 data sets in our database. Which one are there? These ones. This is the name in RECBASE. 15. See? And one peculiarity of this data set that I'm gonna mark in red, like here, they have one peculiarity, and it's that they come from, I'm gonna move myself up here. These 8 datasets are run by volunteers. Another way of seeing these datasets is if I divide them in taxa. Datasets contain birds. We see that there are 5: bird drinking, transects, game, and spring monitor lomus. This one is pretty much everywhere. You see now. Plants, there are 4: vascular plants, spring monitor lomus again, and then forest and restored vegetation, and science, society, phenology. Then we have mammals. Small rodents, Lapland, small rodents. These are the same, but the methodology in Lapland was a little bit different. It is a little bit different, and that's why we decided to make them in a different dataset. Then we have, again, game. The butterflies we have 2, midvas and volunteers. And then moth, we have more moth monitoring, trees, tree phenology, phytoplankton. We have only one, and this is the only one marine. And then we have reptiles, other amphibians, and insects from monitoring, spring monitoring. Okay. If we see this graphically, we'll see that if we count how many data sets have birds, 5. How many have plants and mammal? We have 4. How many butterflies? 2. And then the rest 1. Moth, phytoplankton, trees, insects, reptiles, amphibians, and all. Records. So let's get a little bit deeper into the data. How many records do we have? We have about 6 million records. No, sorry, I said about, no, we have more than 6 million records. And most of the records come from these 4 data sets. You see, this is the name of the data set, and this is the number of records. So, bird ringing, forest understory vegetation, winter game triangles, and moth monitoring account for 62% of the whole database, RECBASE. Unique location. Most of the unique locations come from this dataset. There are almost 200,000 unique locations only for bird ringing, and that accounts for 92% of the, let's move myself again here, of the RECBASE. Let's have a look at species. 70% of the species come from moth monitoring, phytoplankton. You see here there are more than 1,000 species in phytoplankton, science, society, phenology, and vascular plants. Now, time, all these data sets, and then we have here the year from the beginning of the 20th century until now. So let's have a look at, for example, science, society, phenology. What is the time span of this? When did observations start, and when did they finish? Look at this. So science, society, phenology started at the beginning of the 20th century, but no, this is wrong. Actually, this is the longest time series, and this started in 1750. This is the longest, and I didn't put it because otherwise you would see the rest very small. So this is kind of special. This is the longest by far. So what do these colors mean? This color means the records. So the darker the color, the more records. So you can see here the slightly darker here at some point, 1920. Darker here at some point, more records, more observations in the 70s, etc. And I'm going to show you the rest. You see? So, for example, bird transects, it started in 1910 about, and then there were some gaps here, but then after the 50s, 60s, it's been all the time. Vascular plants, for example, this is very special because there were only two surveys in the 40s and then in the year 2000, about 2000, with a lot of records. What else can we see here? Small rodents, this is the same database, but as I told you before, the methodology is slightly different here in Lapland. That's why it's in a different dataset. Bed ringing, there are a lot of records. Forest and the story vegetation is the same case as in the vascular plants, that there are only three, in this case, three surveys. Winter game, summer game, there are more in winter game than in summer game. Most monitoring, there are a lot of records, and this is the shortest time series, butterfly methods. Okay. So now we are seeing the details of the dataset, the records, how many species, etc. And then we have seen time frame, time span, when did observations start in each dataset, when did they end. And now one important aspect, one of the most important aspects, is the location. So let's have a look at the spatial coverage. These are all the maps. These are all the maps. And I just wanted to have a look in general, because I will go through not each one, but some of them. For example, if we separate them in taxa, as I did before, let's have a look at birds. What do we see? What do we see here in birds? We see here in birds the transects, bird ringing, game, triangles, summer, winter, and spring monitoring. This is what I told you before about the bird ringing, that there are almost 200,000 unique locations, so it's pretty much everywhere. There are maybe, sorry, I didn't tell you that these are not dots, these are hexagons, like this, but drawn hexagon, and each hexagon here is 10 square kilometers. So each hexagon is full here. I mean, the whole Finland is full with observations of birds, of bird ringing. So there are maybe some spot here, some spot here in the south. Of course, there are more empty spots here in the north, but the south is completely full. As I tell you, maybe here, it's almost. A winter game and summer game. There are more records here than here in the summer, and the pattern is almost the same, but if you look closely, you will see some difference. For example, here, you see, there are more during the winter than in the summer. Plants, let's have a look at plants. As I told you before, or I don't remember if I told you, but vascular plants is a very local data set. It's only in the archipelago. Then there is this forest and the story vegetation with special pattern in the south and in the north, and in spring monitoring laws and science society technology with a very similar pattern. Mammals, have a look at mammals. Small rodents Lapland. These are only two locations in small rodents Lapland, as I told you before, with a slightly different methodology, and that's why we made them a different dataset. Summer game, winter game again. Butterflies. Now we have butterflies, and very similar pattern. Maybe more records here, and more in Holland here in butterfly midfast. Moth monitoring. And finally, we see a tree phenology and phytoplankton list. There are a lot of records, and of course, only images. And that's it. Thank you very much, and I hope you enjoyed. Of course, if you have any question, just ask me. If you want to have some data, if you want to have some extract of the data, questions about it, just ask me. Thank you.